Your hand is up.
Oh yes, Miss, I'm unable to record.
Okay, no problem. I have more people that I had given the rights. But at
Okay,
least
thank
you're a co-host so that you can hold on to the class so that we are not all kicked out.
you. Okay.
Put your hand down then so that if I have an issue, I know somebody can. Okay, you're able to see my screen, eh?
Yes, Prof, we can see.
I'm trying to close down some programs so that I don't have a lot of interference. So the last time we were discussing muscle fiber types and we were talking about the oxidative, glycolytic, and the way the muscle plasticity, how it changes from one to the other type. This diagram is not very clear, but it captures what we had talked about in this diagram. So you can use the two diagrams to compare and see. So Nicholas and anybody who is a co-host, when you see somebody trying to come in and you recognize them as your classmates, allow them in, please.
Yes,
As
Prof.
long as you recognize them as your classmates, you allow them in.
Okay.
If somebody else trying to come in, just listen. So today I wanted us to start on something else, and this is muscle tetany and fatigue, and the relationship between the action potential and contraction. Now, I think because of what you have here, it's showing you when you have stimulation. What you have here, these red arrows are stimulation. When you have sustained stimulus for a long time, you can have contraction. In the kind of isometric contraction, you see that you can sustain it for a long time. Isometric means that for the same, you're not changing the length of the muscle. Now, when you come to the second one, with sustained stimulation, you find that you may allow this sustained stimulation so that you end up contracting the muscle for a given length of time. It will reach a point whereby no matter how much you want to keep those muscles contracted, you see that you will give in. And by the time the muscle stops contracting, it means no matter how much stimulation, they have reached a point whereby they cannot sustain the contraction. At that point where they fail to sustain contraction, you see that the muscle has fatigued. And there are several causes of fatigue, which we'll discuss at the end at the end of this lecture. The other thing is you need to discuss also about muscle tetany. This tetany is sustained muscle contraction. So what we have here is because of continuous stimulation, you have a sustained muscle contraction, which we refer to as tetany. Now, at the end of that sustained muscle contraction decreases. Now, you can see in this case, when you look at the stimulation, it is widely spaced. The interval between the stimulus is widely spaced, and for that reason, you find that you're able to sustain it for longer. When you increase the frequency of stimulation, you can see then you start to fatigue. If the stimulation period again is increased in frequency, then you can see that they will reach tetany at a very high speed, but then after that, just as fast, they undergo fatigue. So let us see what happens during contraction. I think this is wrongly placed. For those people who have not seen this slide, it is just showing you the hypertrophy of muscles. I think I had already discussed this. So I wanted us to start on the muscle tetany, which is sustained contraction of a muscle as a result of rapid, successful nervous impulses. So you have continuous sustained stimulation, and the stimulation is coming from the nerve. So as long as you keep on stimulating that muscle, it will keep on releasing the calcium. Remember what we discussed about excitation, contraction, coupling. So as a recap, the nervous stimulation will cause release of neurotransmitters. Neurotransmitters will bind on the motor end plate. Motor end plate will cause depolarization, which may pass to the sarcolemma. From the sarcolemma, it will infiltrate to the T-tubules. From the T-tubules, it will cause opening of calcium channels in the ER. Now, this calcium will flood the sarcoplasm and bind to the troponin on the calcium binding site and initiate the sliding filament mechanism, leading to contraction. So, since you can see all that part, it means that if you keep on increasing the frequency of stimulation, you are going to have that sustained depolarization of the sarcolemma, and because of that sustained depolarization, you keep the sarcoplasm flooded with calcium. And as long as you have calcium in the sarcoplasm, you're going to sustain contraction. But you can only do it for a limited period of time, because after some time, you find that the muscle cannot sustain this any longer, and therefore they undergo fatigue. So let us look at the steps of formation of tetanus. Now, if you remember, there was a slide I was showing you, which was showing you an action potential. So if we look at action potential, and I think you have even done this in the lab. So if you look at the lower graph, you are looking at muscle tension. And when you look at the upper graph, you are looking at action potential. So if you have, in this first graph, you have a depolarization. So if this is the resting membrane potential, so you'll have a depolarization, hyperpolarization, and going back. And then you have rest. When you have this action potential, it goes. Remember, we have a lag phase. The period between the time you stimulate motor end plate, because from the nerve, it has to go through all those sequence of events. So you find that contraction does not start immediately. You start having a depolarization. From the time of stimulation, it will take some time so that those steps that I have mentioned will go through. So by the time you have calcium being flooded, you are a bit off. And when you start sustaining contraction, or rather when you start initiating contraction, you'll have, for one stimulation, one single muscular contraction, where the tension increases and then goes back to where the muscle was at rest. So this lower line is showing you the muscle at rest, This other one is showing you, the upper graph is showing you the stimulation. And this is similar to what you are seeing when you went to the lab. So using the Power Lab, it would show you the point where you clicked on the button to show you initiated the stimulation. And then the graph would show you the point whereby you start seeing muscular response. So the interval between the time you started the stimulation to the time you start seeing muscular contraction, this is what you call the lag phase or the latent phase. So now, a single response to a single stimulation is what you have here as a switch. So here you have your stimulus, the lag phase here, and then you have increased muscular tension. Now, in the second one, you have the first stimulation here. And what will happen is that following the same events, you will hit a peak, and as the muscle starts to relax because the stimulation is over, and therefore you are going to end the depolarization. And when you end the depolarization and the calcium has been pumped back into the sarcoplasmic reticulum, you find you don't have calcium in the sarcoplasm, you start to relax the muscle. But before it has relaxed, you set off So can you hear me?
Yes, Prof.
Oh dear. Today will be a tough one. Let's just see how far we can go. So as before I was kicked out, I was showing you that in this diagram, the amplitude of the peak is at that point, but in the second one, the peak is higher. So my question was, who can attempt to answer as to why we have this increase in peak? And you can see even with the second one, if you initiate another stimulation, the peak is even higher. Why do you think this is happening? We have like a step ladder. Remember, you may not have the correct answer, but at least it's an attempt. What would cause the second or the subsequent peak to be higher? Sebata, unmute yourself.
Is it relative refractory period? Like it takes a much stronger stimulus to get the thing?
Then in that case, then it should be even smaller because you need a stronger stimulation to reach the original peak. Then if you are to use the argument of refractory period, it would be the opposite.
Okay.
Okay, Nicholas, let's hear yours.
I think more calcium are flooding the sarcolemma. So
There is a lot of flooding. Who is the other person who put up their hand and then they put it down? Where is my other pharmacist, the confident one, who had his hand or her hand up? I don't know who it was.
It's Grace.
Okay, Grace, what's up?
I wanted to say what Nicholas just said.
Yes, so we have, remember, if this is the myofibril and you have flooded it with calcium, and this calcium needs to be put back into the stores. These are the ER. So if you want to put back this at this period of time when you are relaxing, what you're doing is you're removing the calcium and pumping it back into the ER. Now, before you have managed to pump it all up so that the muscle completely relaxes, we still have some calcium in here. And then you flood it with more calcium. So your baseline is higher than it was initially. So if you imagine a relaxed muscle, in this relaxed muscle, you did not have calcium in the sarcoplasm. And therefore, when you flood it, you are starting at a low baseline. If this is the concentration of calcium on this side, so we are looking at calcium concentration here. With the first stimulation, you are starting when the calcium is low here. So you flood it into the sarcoplasm. And then when you are trying to return it into the ER, you initiate a second stimulation. And before this calcium has gone back to zero, you still have calcium flooded in. So now you bring in more stimulation. So you are starting from these points. That means the amount you've added will add on to what you initially had, and the peak will be higher. If you start to relax again, and before it has gone back to where it was, you stimulate it, then you have a higher amount of calcium in the sarcoplasm. Now, the amount of calcium in the sarcoplasm is the one which is determining the level of overlap, because you still have a lot of troponin bound to calcium and therefore forming cross bridges. Those cross bridges are the ones which initiate contraction and therefore create tension on the muscle. So before I move on, anybody who has a problem with that? Anybody who did not understand that component, why we have this step-like increase in muscle tension? Yes, Lojore, at what point did we lose you?
Okay, so my question is, when you add, you're telling us the amount of calcium affects that contraction, right?
Yes.
So when the calcium is at the point you've shown us in the second diagram,
Mhm.
it means the amount is still like the amount it was before. You just added more to increase the contraction, right?
You just flooded it with more calcium.
Oh, okay, that's what I wanted
So
to
when you flood it, remember what you have here on this lower graph or the horizontal axis, it is representing time. So with time, muscle tension is increasing. What is causing the increase in muscle tension is you are forming cross bridges, and you form until you reach the time of maximum tension. And at that point, you start removing calcium from the sarcoplasm. If you remove calcium, it means that the bound calcium, which was bound to troponin, will be released. When you release, the muscles can no longer form the cross bridge, the cross bridges, and therefore, what will happen is with time, the number of cross bridges will reduce, and when you reduce cross bridges, you reduce the tension, so the tension starts to drop. So what you have here is actually muscle tension, but the result of this muscle tension is as a result of calcium in the sarcoplasm. The more calcium you have, the more troponin you have bound. And when it is bound, then it forms more cross bridges. When there are more cross bridges, the muscle tension is higher. So you can see the relationship between calcium and muscle tension. Okay?
Yes, miss, thank you.
Anybody else who is lost at that point? Because what we are going to do, you can see that in this case, it was a single stimulation giving you a single twitch. Now this one, it shows you that that muscular contraction wave is actually the second one is being added to the previous one. And that is why we are calling it summation. You are summing up the wave or the muscular tension. So you sum up by adding on to what you previously had before it completely relaxed. Now what will happen is that this period of relaxation, this intermittent period of relaxation, if you increase the frequency of stimulation, you find that this time will become even less. And as it becomes even less, it means that now the wave starts looking like it is a continuous wave. Instead of these big step like ladders, now you can see between here and here is a very small step. Between here and here is a small step. So generally, if you look at it, it looks like it is a continuous ladder, but a continuous incline, but with teeth projecting out. Now, when you increase the frequency, even these small indentations, they merge up. And what you'll end up is having one large continuous wave. By the time you have this continuous wave without this teeth-like formation, then you say you have complete tetanus. Tetany is that state of continuous muscular contraction. So this one, between this twitch and maybe another twitch here, then there is no continuous muscular contraction. But when you start to add up a wave to the other one, what will happen is that with increased stimulation, this wave will start to fuse. They'll start to merge together. But as long as you can see this teeth-like projection, so you still have that tetanus. However, because you can observe this, then you call it the incomplete tetanus. This is as opposed to when you have that continuous sustained contraction here without those teeth-like indentations in between the stimulus. So you move from a twitch to a wave summation, incomplete tetanus, and then complete tetany. Now, I'm sure most of you have heard of a disease called tetanus. When you have tetanus. Now, what happens in such a case? You have a viral infection, and that virus will initiate continuous stimulation of the muscles. So it is a virus that gets into the nerves, initiating continuous stimulation of muscles. Now what will happen is that your muscles will all, because of that continuous stimulation, they will crunch and they will actually lock up in a contracted form. And in fact, when your facial muscles contract like that, the jaw loses its position and it's trying. What happens is that it clenches the upper and lower jaws. They lock together and you cannot even open the mouth of the person because of this continuous tetany of the facial muscles. For that reason, another name given to the sickness of tetanus is lockjaw, because your jaws are locked and you can't separate them. So you find that in some of the textbooks, they'll ask you, lockjaw is a result of what? And you're supposed to explain, because you should know what lockjaw is. It is that condition of tetanus. So it is important for you to see how the tetany builds up. So now, remember we talk of a refractory period in the neuron. That refractory period is that period that you need rest, during which time you replenish whatever you had used up. And during this time, you find that it is difficult to stimulate. If it is a neuron, it is difficult to stimulate the neuron to the same extent. Now for muscle, we also have a refractory period. During the period whereby you are trying to replenish, you are trying to put back the calcium where it should be. So this period is what we are calling the refractory period of the muscle. Now, during this time, even if you try to contract the muscle further, it is difficult. So it has to at least have gone a small refractory period during which now you can initiate a second one. So you find that when you have the refractory period is so small here, if it is beyond the refractory, if you stimulate anywhere after the refractory period, it means that you can initiate a second wave. So you can see in this case, at this point, the second contraction falls after the refractory period, which is here. Then this one will also have its own refractory period, and the third wave comes after the refractory period. Now, what happens is that when you have sustained contraction and you're not allowing that refractory period, is when now you have the merge of the wave and you have continuous tetany. Yes, Lillian.
I wanted to ask,
Yes.
Is it true to say that rigor mortis is an example of a tetany?
Now, rigor mortis, continuous contraction, but not due to muscular stimulation. It is continuous contraction due to lack of ATP causing detachment of the myosin heads. So in this case, this is continuous stimulation of the muscles causing the myosin heads to keep on having those cross bridges. They release and bind, release and bind. So you are still having cross bridges and ATP use up during tetany. In rigor mortis, you cannot use ATP, you don't have ATP, you cannot detach. So the two physiologically cannot be compared. They are not actually the same. Lojore, you have a question or it was a previous one?
Oh, sorry, it was a previous.
Okay, so I lower your hand. Mustaf, Eden.
Yes, Prof. I wanted to ask about the concentration of calcium in the cytosol. Like most of the time, this concentration is due to a depolarization or a stimulation of the muscle. So I was asking, is there any other condition that will ensure elevated calcium concentration in the cytosol apart from stimulation? Maybe kind of like maybe some disorders or something else like that.
Oh, definitely. Remember when we were looking at calcium concentration? You remember when we did that? You did some presentations, I think, regarding what is it called? The electrolytes and electrolyte balance. Yeah. And we said there are conditions whereby you can actually have abnormal amounts of calcium, and what they'll end up doing is affecting the neuro and muscular activity.
Oh, okay.
So, under normal circumstances, we have the optimum amount of calcium that you can have in a cell, because calcium is not only used for muscular contraction. You also use calcium as a second messenger for other processes. So you still may have some calcium, but required to run other processes. But when you flood, now you have up and above that normal value that you usually have. So if intracellular calcium is, say, 0.4 or 4 millimolar inside the cell, and now you stimulate it and it goes to 5 millimolar or to 6 millimolar, you see it is above what you normally have. And that above component is the one which is causing these other processes, additional processes like muscular contraction.
Okay, thank you.
All right. So there is no other question, so we move on. Now, in this, this diagram shows you the refractory period. Remember we said when you cannot initiate another contraction because it will not go, from here it can only go higher than that, or the period of time from when you started the contraction to when you start relaxing, repolarizing. So now for the skeletal muscle, you can see that it is a continuous peak, and then from there it starts to relax. The refractory period in cardiac muscle is much, much longer. So the cardiac muscle will contract, it will reach a peak, last long, and then after a period of time, that's when it starts to relax. And you'll see when you're looking at cardiac muscle cycle, is that when you initiate stimulation of cardiac muscle, you flood the cardiac muscles with calcium. There are two types of calcium channels. There are those slow, and then there are those long-lasting. They last longer than the normal period. So when you open those, the long-acting channels, they'll keep the calcium flooded in the sarcoplasm of the cardiac muscle for a longer period. So initially we have, like for the skeletal muscles, the normal calcium channels which open continue, and immediately we have a flood of calcium in the sarcoplasm. But then we also have the longer acting channels which open up later in cardiac muscle, allowing the calcium to stay prolonged inside that cell. For that reason, the cardiac muscle, the contraction period lasts longer than the one of skeletal muscle. I think this one was trying to show you, oh yes, smooth muscle contraction. I think I have gone beyond what I wanted because I want to dedicate a lecture for smooth muscle contraction and another one for cardiac muscle contraction. So I will not do it now. For today, I wanted us to look at the energy sources for muscular contraction. Oh dear, where am I? So I think I have two sets of slides which are talking about summation. So these are the ones that are just giving a summary of what we have talked about. When you have muscle tension increasing with increased stimulation. So in the other one, we are showing when you have increased frequency. Remember, you had talking about increased frequency. But the other time when you were in class, we were talking about increasing the strength of stimulation. So when you stimulate a muscle and you do not reach threshold, that muscle will not contract. However, when you reach threshold, you only recruit the first motor unit, and then from there, you're going to recruit more and more and more motor units until you have recruited all of them. Then in this case, you have all motor units responding to that stimulation. So this graph is slightly different from the one that we were showing before of muscle tetany. This one is showing motor recruitment. I think we did that in class. So this is where we were talking about the multiple wave summation. I've already discussed this, showing you that the point of stimulation, but the other one was on the other side, on the lower side. So this wave is just showing you similarly to what I have shown you in the previous diagrams, only that this is shown in a different format. Yes. So the other component I have not mentioned is strip or the graded response to a stimulation after a sustained long period of rest. Yes, Anastasia, you have your hand up.
Yeah, I have a question. There's something I can't understand about the frequency of
Yes.
the, okay, is the frequency the same as the strength applied to the muscle? Like, for example, when I'm carrying a weight of 5 kgs and then you add another one for 5 kgs to sum up to 10, is that the, okay, is that what you're calling the frequency, or it's the?
No. When I have 2 kgs, I will stimulate my muscles to respond to that weight so that the tension created will only oppose 2 kgs. But if I double the weight that I'm holding to 4 kgs, what happens? You have to send a stronger stimulus to oppose the added weight. Okay? So when you first sent the stimulation towards 2 kg, you recruited a number of motor units. And those motor units are the ones which stay contracted to oppose the force of 2 kgs. When you add another weight, it means in addition to the motor units you had recruited before, you have to add up more motor units so that the additional motor units are going to support the additional weight. So what you have done is increased the the motor unit by increasing the strength of stimulation. So what you have done is you have tried to increase the strength of stimulation so that you recruit more motor units.
So how are you increasing the frequency there?
Okay, so now for the frequency is when I send, I'm sure all of you have gone through this, whereby you just feel something like a rush go through your muscle and you say, wow, that's a twitch. It means your neurons sent a single stimulation and your muscles made a single contraction in response to that stimulation. That is a twitch. However, when you want to move your hand, okay, when you want to move your hand, you cannot rely on twitch. You have to contract that hand. So what are you going to do? You may not increase strength, but what you are going to do is to increase frequency. So now, let's say your hand is holding those books. That's a weight. For you to keep it sustained in this level, it means you have to keep sending a stimulation. Keep sending it. If you stop sending the stimulation, the muscle will relax and your hand will straighten and your load will fall. So to prevent this from happening, you have to keep sending stimulation to keep this muscle contracted. That is increased frequency. Now, Increased strength is if I increase not the frequency, but the strength of stimulation. I'm going to recruit more motor units here. So if I had recruited 5 motor units to keep this hand here at that level, I had made sure I have recruited 5 motor units. If I increase my strength of stimulation, I'm going to recruit maybe 7 motor units. If I recruit 7 motor units, it means that this muscle will increase the tension. If it increases the tension and the load remains as it was, so I still have, say, 2 kgs here, but I have increased the strength of contraction. So I have recruited more motor units. What do you think will happen? It's a question, Anastasia.
Yes.
If initially, to keep my hand horizontal like this, so that I'm holding a weight of 2 kgs, I need to recruit 5 motor units. You understand that part, eh?
Yeah, I do.
Let me increase my strength of stimulation. That means I send more firing here so that I end up recruiting 7 motor units. If I recruit 7 motor units here and I still have the 2 kgs here, what will happen?
I think you lift it.
You lift it, isn't it? It will contract more and therefore you'll end up lifting the load, isn't it?
Yeah.
Okay. Imagine I increased this stimulus strength, but I also increased the weight here to 5 kgs. So now what I have done is I've recruited more motor units to sustain that 5 kg at that position. So can you see that the muscle length has not changed?
Yeah.
Because I increased the stimulus strength, I recruited more motor units. So they still kept my hand at the same length. Okay?
Yeah.
You increased the strength, but you are not changing. You're not changing the length of the muscle. You are still keeping it there. So the force it is opposing is much more. For it not to hold upwards, it means that you are putting a greater weight here. So what you have done is you have increased tension on this muscle. You have
I understand
increased,
that.
yeah?
I get that, but I'm asking, what's the essence of changing the frequency?
Why you want to change the frequency?
Yeah, but like you've been explaining about the strength, change in strength and recruiting of the motor units, but now the frequency.
Why you need to change the frequency?
Yeah.
So now, this brings back to the sensory physiology. You remember coding?
Yeah.
And how do neurons code for information? When you change the strength of the stimulus, what do you do to the frequency of stimulation?
Increase?
You increase the frequency. That's where the frequency issue is coming. Increase the frequency of stimulation, you'll end up increasing contraction or tension. However, in this case, when you are increasing that frequency, it will reach a point whereby there is the maximum tension you can develop in the number of motor units that you have. Yes, Asna?
I also had a question. I wanted to ask if you have, for example, the 5 kg load and a 2 kg load, the 5 kg load, since it's stronger, it will release more, it will have a higher frequency.
Come again, if the 5 kgs does what?
Since it's the load is stronger, will it fire more frequencies?
If the load is stronger, will you have usually? Now it's taking me back to coding. You remember, you first recruit from the same neuron. You first increase frequency so that you can show the strength. When you increase the strength, you increase the frequency in the same unit. But when it reaches a maximum frequency for that unit, you have to recruit more units. So you have to go to the next level. Do you remember that? But we were talking in that time, we were talking about sensory information. So I remember I had given you a diagram where we have sensory inputs here. We have a sensory ending here. And we have a sensory ending here. All these belong to one sensory unit. We are taking information there. But then we also have a sensory unit here, a sensory unit here, and another one here. And these are taking information to the second group. Yeah? So initially, if the strength was enough to stimulate all these, you find that the interval between stimulation here is spaced. But when you increase the stimulus to recruit the second sensory input, then the frequency increases. But then if you increase again and you stimulate the third sensory ending, you find that this neuron might reach its maximum. You can't increase frequency more than that for this neuron. So what do? Because you are still coding for information, you have to inform the body. The strength is increasing more than it was before. You start to recruit the second unit. So now we'll have this one at its maximum and this one also sending information. When you increase the strength, again, you double up the frequency here. So this was for sensory input. We talked about coding regarding sensory inputs. Are we together?
Yes.
So now reverse the situation and put it for the motor unit. Motor unit is one neuron and all the muscle fiber it innervates. This was a sensory unit. So now I need a strength to hold 2 kgs. Although I have many motor units here, I can have the blue and the red ones here. So if the red motor unit is only causing contraction of these fibers, and this one is strong enough to support 2 kgs, then I don't need to recruit the next motor unit here. But if the 2 kgs are sustained by the red, and then I add, and now I have 5 kgs, the 2 kgs cannot be supported by the red fibers. So now I have to recruit the second fibers. So I recruit a second motor unit. And the more weight I increase, I find that I need more motor units so that I can sustain the increasing weight. Okay? At least you've understood that part, yeah?
Yeah.
Okay. So now, let's assume I have 2 kgs So I am not going to recruit the black one. I'm going to send a stimulus, a single stimulus to the red units. A single stimulus to the red unit will cause a single twitch. And therefore these muscle fibers will contract and then they'll relax. And if they relax and my 2 kgs are still there, my 2 kgs will drop. So to prevent my 2 kgs from dropping, after I send that first stimulus, I have to send another stimulus so that it keeps on holding on the 2 kgs. Now, if I don't send them frequent enough, what I will end up having is my hand swinging between interval. So between the first stimulation, my hand will drop. And then I send another stimulation, it goes back to where it was. If I don't send another stimulation, it drops. So to prevent that swinging of my hand, I have to make sure I send enough continuous stimulation to the same motor unit. So what I'll have done is increased the frequency to this motor unit so that I sustain the same amount of tension to hold 2 kgs. So here I have increased frequency. Here I have increased by recruiting more motor units, I'm increasing strength. Now you see the difference.
Yes.
Anybody else who is lost there? Yes, Nicholas.
Yeah, so my question is, so can you conclude that frequency is the number of contraction like made within a period of time, and also intensity of stimuli increase the frequency, and that also decrease the time of contraction?
No, because I want you to separate the two things. Frequency is the number of stimulations per given time, regardless of contraction. The muscle will respond to either strength or frequency. The muscle will respond to both. So when you talk of frequency, you're just looking at the same amount of units you are stimulating. You are not increasing the number of units. It is for the 1 unit, and you're looking at how many times are you stimulating. So I have my red motor units. For the same motor units, how many times am I stimulating them? So I will look at the frequency of action potential in the same motor unit. So I'm looking at stimulus per given time. When I increase the strength, I recruit more motor units. That will ensure I have enough tension to support more opposing force. This one you don't need to add more units because it's only a small weight. But if you don't sustain the stimulation, even that weight, you cannot sustain it. It will drop. But this one, you need to recruit more motor units. But then after you recruit those more motor units, you also have to sustain the stimulation to keep it contracted. Because if you stop the contraction, even that greater weight will fall. So you have to separate the two things, frequency and strength. Is that clear, Nicholas?
Yes, Prof. Thank you.
Anybody is lost? Okay, so now we have a muscle which is at rest. You find that when this muscle is at rest and you stimulate it, it will reach a peak and then it will relax. But the peak, because it is coming from like ground zero, it will only manage a given level of a peak. Even if it completely relaxes to where it was, the original length, you find that if you stimulate it a second time, the second peak will be much higher. And remember, it is rising from the ground zero. The second peak will be much higher. If you are to allow it again to completely relax and then you stimulate it a third time, the peak will keep on increasing. So the tension is rising with time, so that finally it will have a maximum tension with every stimulation. So if you look at the level of tension, it is a graded response, but increasing until it reaches a maximum. And then from there, it now flattens. At this point is when you increase the frequency, you start seeing the merging of the wave. But when we have complete relaxation intervals, then this is not similar to what we had seen before, which we were calling the incomplete tetanus or wave summation, because the waves have not really merged. But we see the step-like ladder increase in strength or amplitude of the contraction. This is what we call the trip. And this one usually occurs after a prolonged period of exercise. So you find that when you start trying to stimulate, your muscles are not responding as they would if you are continuously activating those muscles. So it usually is a response that occurs after a prolonged period of rest, and then the the subsequent contraction becomes stronger and stronger till it reaches a maximum. Now, by the time it reaches a maximum, then it will be very easy to contract that muscle. So this diagram is a summary of what we have just discussed, but all compiled in one diagram showing you from the twitch, latency period, contraction period, summation, incomplete tetanus. For those who want to read, they can read more on this. So I think from there, the next thing I want us to look at is the sources of energy for contraction. What are the different sources of energy for muscular contraction? Now, when you look at muscle contraction, we talked a lot about how it requires ATP and how ATP can be depleted. And when the ATP is depleted, cross bridges cannot break, and therefore you have continuous contraction in case of rigor mortis. But to prevent that, you have to have a continuous supply of ATP. So what does the ATP do in the muscle? Here it is summarized that it is required for attachment and detachment of the myosin heads to the actin filaments. That is the cross bridge formation. Number two, we use a lot of ATP for the calcium pump. Remember we talked about pumps and we said pumps have to use a primary source of energy, which is normally ATP. So we require ATP to return calcium back into the sarcoplasmic reticulum. Number 3, we need ATP to fuel the pump, the sodium-potassium pump, which can restore membrane polarity. So it is not just ATP inside the sarcoplasm. It will go all the way to even to the sarcolemma, so that it can maintain membrane polarity, or it can take back the membrane to its resting membrane potential. So for that reason, we need a lot of ATP in the muscles. And you find that the ATP which is already flooded within the sarcoplasm cannot last long. On average, 4 to 5 seconds or even 6 seconds. So on average, talk of 5 seconds. And you've depleted your cytosolic ATP. Now, if you can deplete so fast and you are doing muscular activity, where are you getting this ATP from? It means that you have to keep on generating ATP, and our main source of ATP is usually we usually have several sources. First, after we deplete it, we have the source of ATP store. Now, you've all heard of this molecule creatinine. Creatinine acts like the way myosin acts by holding on to oxygen. And when you need that oxygen, the myosin has to let go. Sorry, not myosin, the myoglobin has to let go of the oxygen. So it is acting like a temporary storage. Similarly, creatinine is a molecule that you find in the muscles, and it acts like a temporary storage for ATP. How does it do that? Because creatinine is phosphorylated by ATP so that it forms creatinine phosphate. Now, when the need arises, this creatinine phosphate can actually phosphorylate an ADP to generate back the ATP. And now the ATP will be available for use. Are we together up to that point? It's a temporary storage. How does it do that? When the muscle is at rest, you don't require a lot of ATP. The ATP that is being generated is continuously being used by the pumps and by these other things. If that happens, it means that when you need to contract the muscle, you'll have depleted your ATP from the use of the pumps, from the use of calcium pump, sodium, potassium pump, so you don't have enough to cause muscular contraction. So in order to have an available source, immediate source of ATP, creatinine acts like a temporary storage by being phosphorylated during rest by the ATP so that you have creatinine phosphate. Now, when you need to contract the muscle, you find that the ATP in the cytosol will be all used up, forcing the creatinine phosphate to phosphorylate ADP and replenish the ATP so that you still have enough to continue the contractile process. So remember, the first one we said can only last about 5 seconds. So if you are to use the ATP in the cytosol for contraction processes, they can only last about 5 seconds. By the presence of creatinine phosphate, you can prolong the period of contraction up to 15 seconds. So now you have raised from 5 seconds availability of ATP to 15 seconds availability of ATP. So I will show you this summary so that you can understand what I'm talking about. You have creatinine here. Is it this one? Creatinine can be phosphorylated by ATP to form creatinine phosphate and ADP. The enzyme responsible is the creatinine kinase. Remember, what does kinase do? A kinase is an enzyme that phosphorylates another protein. Our molecule here being phosphorylated is creatinine. So once it is phosphorylated, you end up having creatinine phosphate and ADP in the cytosol. Now, when you need the ATP, you have to break down this one so that it releases creatinine and ATP. And now your ATP is available for use for contractile process, which can last up to 15 seconds. Now, if you are to go beyond that, if you are to go beyond the 15 seconds, then it will depend on what level of activity you are doing. If you want continuous, sustained production of ATP, then you have to go through the aerobic pathway. Generate using oxygen. However, if you need a burst of energy, a burst of resource of ATP, and you don't have enough oxygen, then you have to go through the anaerobic pathway. Now, whether you are using aerobic or anaerobic pathway, the first step will be involve the anaerobic pathway. Why? Because if you look at the process of glucose utilization, it first undergoes glycolysis, whereby it usually releases 2 pyruvic acid plus 2 ATP molecules. And I think I mentioned that in class. So we first have the glycolytic cycle, and it only produces a little amount of energy. Now, if you add this energy that is available, then it can take longer than the 15 seconds. It can double up the time up to even 30 seconds. So you can have that burst of energy available lasting for even 30 seconds, and you are okay. You don't even need to oxidize the glucose. The problem with that is that now you will be having a byproduct, which is pyruvic acid. The problem with pyruvic acid is that it cannot diffuse out of the cell. It is stuck inside the cell, and then it is highly toxic. So what your body has done is to manage to have enzymes that will convert pyruvic acid to lactic acid. That way, it will produce a less toxic acid which can easily diffuse out of the cell. So the lactic acid that is formed from here can actually go into the blood circulation, and it is taken to other organs which can break it down. And I'll come to that later. So for now, what you have done is using glycolysis, you are providing the muscle cell with ATPs, 2 ATPs in addition to what you already had. So you first have the ATP that has been created is free in the cytosol, you've used it up. Number 2, you start using phosphocreatinine to generate ATP, and now you've used it up. Number 3, you start using the glycolytic cycle so that it produces an immediate source of energy, ATP. The problem is that immediate source comes with a price, and the price is formation of acid. Now, we'll come to the muscle fatigue, and you'll see that one of the things that have been attributed to fatigue will be the buildup of the lactic acid and the depletion of other electrolytes, but we'll come to that later. Now, from there, I've already said that from there you'll end up having to go into the aerobic pathway. But I've already mentioned that the problem with this, you end up having a lot of buildup of lactic acid. And the problem with this, it cannot last long. It can only go for about, for about 30 seconds. The only advantage is that it has an immediate source of energy. It is faster than going into the glycolytic cycle, not the glycolytic, but the Krebs cycle, and it does not require oxygen. So you don't have enough time for the oxygen to go into the muscle and to go through the Krebs cycle to produce the ATPs. For that reason, you generate an immediate source of energy and you have enough to do what you want within a very short period of time. So you find that during exercises, when you require to utilize like the slow fibers, not the slow fibers, but the fast fibers, when you just lift a given weight for a short period of time and you need massive amounts of ATP to provide the tension, you don't have enough time to oxidize. So this will be the pathway that you usually provide the energy for the contractile process. So before I move on to the aerobic one, do I have any questions so far? Or am I moving too fast? Yes, Nicholas.
Yeah, I have a question and it's not on this one.
Yeah.
It's on the when you were explaining creatinine. I was wondering, like the inorganic phosphate release when ATP is broken down into ADP and inorganic phosphate. That inorganic phosphate, can you also bind to creatinine to form creatinine phosphate?
No, because for the creatinine phosphate, you require ATP, and the ATP is the one that is providing the phosphate. Remember the way enzymes work. They have a pocket whereby they put a given substrate. So you have your creatinine kinase here. So it will have a pocket. It has to have for ATP and it has to have for creatinine, so that it brings the two together and releases the two molecules. So if that pocket cannot fit the inorganic phosphate, it cannot catalyze that reaction. So it has to take a given substrate, which is ATP, and the other one creatinine, so that it can generate that. Okay?
Yeah, as a follow-up question, so what is that inorganic phosphate used for?
Inorganic phosphate is used to generate ADP. Remember when you are trying to form ATP.
Yeah.
ATP, you have to add a phosphate group. So the inorganic phosphate is used to generate phosphates, and the phosphates are the ones which are going to be added there. Remember, in the cytosol, it's not like you have some marked inorganic forces which has to follow this path, and some other phosphates that are marked green. Let's say you cannot mark some red and say these ones are used to generate ATP, and then you mark others green. These ones are to phosphorylate proteins, so that you have the protein phosphorylated proteins by the kinases, so that they form something else. It is, you have a pool, you have a sea of inorganic phosphates. Are they available for the individual processes that require inorganic phosphates? So it will take you back to the first lecture where we were discussing electrolytes and their uses. And we said we need phosphates in the cytosol. And the reason why is because we need the phosphates to generate ATP. We need phosphates to go in the nucleus. We need phosphates to phosphorylate proteins. The phosphates you cannot form unless you have inorganic phosphates. So you see the connection. Don't compartmentalize even the cell. It's a pool and you have all these forming the phosphate, and then the phosphate is available for other uses. Okay?
Yeah, thank you.
I think now I will give you a break, a 10-minute break, so that you come, we finish up, we summarize on energy uses. Then I will give you time to review. I will ask quiz 2. Remember, quiz 2 is coming on Thursday next week. I will ask all questions from the beginning of half semester all the way to what we have covered. Did I include Tuesday? No. Because Tuesday, I'm assuming people are back. We are supposed to meet at 9:44. According to my phone, I'm actually 3 minutes late, so everybody should be in. So I think I will start. I was looking for this diagram, which I knew that I had in my slides, which sort of summarizes what is happening inside the cell. Now, we started off with an energy source being ATP that is flooded. This is a myofibril representing the muscles. This is a mitochondria. This is where we have aerobic respiration. Now, for aerobic respiration, the substrate could either be the free fatty acids, which are usually they're being delivered by the blood. They get into the cell, the mitochondria, and using oxygen, which is also being provided by the blood, they undergo glycolysis and they release ATP. In addition, we also have glucose coming into the cell. But again, glucose can be used as a fuel and provide ATP. But in most cases, when the muscle is at rest, we do not utilize glucose as a source of fuel. Most of the fuel is coming from free fatty acids, or in other cases, you can have, if you don't have the acids, you can actually even start depleting the amino acids resource. But you don't go to that extent unless you're going through the starvation. That's when you start going through the other sources of aerobic respiration through fatty acids and what is it called? Proteins. Now, when we have ATP being generated, we have a flood of ATP in the cytosol. That ATP will be used in addition to sodium potassium pump, in addition to the calcium pump. It will be used for phosphorylation of the creatinine so that you have phosphocreatinine. But it also is utilized for the buildup of the glycogen molecule. And we know that glycogen is a repeated units of glucose. So we need ATP to generate that hydrolytic process, not hydrolytic, it is the process of the glycogen formation. So you find that in most cases, the ATP is being utilized for other purposes. So we have very little amount of free ATP. And that's why if you are to contract that muscle, the ATP that is available can only last about 5 seconds, if not really in advance. So now, the next step is when you have to utilize more than what is available, then you have to start burning down. And I think we had already gone through this slide where we were saying you can use the glycolytic pathway, but it ends up forming lactic acid. I had another slide that I wanted to show you, this one. So it may look very similar to what I had before, because I have a myofibril and then I have the blood vessel here. The blood vessel is providing oxygen and free fatty acids. Now, when you increase your activity to more than the resting stage, your immediate source of energy, because you'll have depleted the ATP in the cytosol, now you have to utilize glucose. And the glucose, because you didn't have a lot of it, because all the glucose that was coming in during rest was being stored to glycogen. Now you have to start breaking down this glycogen to provide glucose. And then that glucose has to start undergoing hydrolysis so that it can generate ATP. So the first stage here will be the anaerobic process. However, we have two pyruvic acids which are generated from this process, and they end up getting into the Krebs cycle. Now, it is from this Krebs cycle that you're going to have the aerobic process, which is utilizing the incoming oxygen. Okay, so by the time you are doing this kind of activity, you are still able to utilize the fatty acids and the oxygen coming in to generate this one, ATP. Now, if you are to increase further, this activity, then what you're going to cut short of here is oxygen. So what I wanted you to note between this slide and this slide, can you see what the blood is providing? At rest, you have glucose, which is being taken into the cells. You have oxygen, which is being taken into the cells. You have free fatty acids, which are being taken into the cells. Only that these two will be used to generate ATP, but this one will be used to convert to glycogen. Now, in case of moderate activity, whereby you have some oxygen coming in, but you don't have enough glucose, then you start utilizing your glycogen stores, and that's where you have diagram. Am I going backwards or once? Okay. So you'll end up having this diagram, this one, where you'll have cut off the source of glucose because you don't have enough being replaced or taken into the cells. But because you still have some oxygen, it still allows you ATP formation. And what you're going to also utilize is the glycogen stores. So you still have the glycolytic cycle and you still have the anaerobic cycle. Now, when you require a burst of energy, you don't even have enough oxygen. What we'll end up doing this one. What you'll end up doing, you'll have cut off the oxygen, you'll have cut off the glucose. So now, where are we getting our energy from? Because we do not have oxygen anymore, our only source of ATP will be this glycolytic cycle. So it will end up having a buildup of pyruvic acid, and this pyruvic acid will be converted to lactic acid. And this lactic acid is taken away by the blood vessels. So now we are moving from one source of energy to a different source of energy to a different source of energy. So I just wanted to show you these diagrams so that you keep them in mind. Remember, In the case of aerobic respiration, we have mitochondria here. We do not have buildup of pyruvic acid. The pyruvic acid gets into the mitochondria, and the mitochondria generates ATP. Now, you find that when you're doing prolonged exercises, those like the Kipchoges we are talking about, endurance, where you are utilizing oxidative fibers, then you do not go into this category. It will still be high activity, but because they are endurance exercises, you have already trained your body to keep that sustained supply of glucose. Then in that case, you will end up having the pathway, this one that requires oxygen use. This one that requires oxygen use, and you end up having a lot of ATP being produced. So when you're talking of moderate exercise, it is either you're doing some exercises where you are able to replenish the oxygen, or you're having those endurance exercises which allow a continuous supply of these two. So now that I have shown you the diagram, let me go back to where we left at. I think we were at this slide when we left for the break. So you find that with anaerobic, the one that does not utilize oxygen, it's mainly glycolytic, and it ends up with lactic acid formation. Now, what happens to this lactic acid? I say the first thing you have to remember, it diffuses out of the cell, which is not possible with pyruvic acid. Number two, once it diffuses out, it gets into the bloodstream. What is the destination? It will usually end up in the liver, kidney, or the heart. These three organs have the enzymes capable of converting pyruvic acid back to lactic acid to pyruvic acid, and this pyruvic acid can get into the mitochondria. So you find that in addition to the other sources of energy, these three organs can utilize the lactic acid that is available to provide the ATP. But how do they do that? By first converting it to pyruvic acid, and then the pyruvic acid can then get into the Krebs cycle via the mitochondria, and from there they yield their energy source. Now, if you compare how much energy is being produced by the Krebs cycle versus by the glycolysis, you find that the glycolysis or glycolytic cycle only takes care of about 5%. Now, if I were to ask a question, instead of giving 5%, I give 95%, then you know that 95% will be aerobic respiration. The only advantage I said to this type of respiration is that it produces ATP so much faster than using the Krebs cycle. Number two, you don't need the oxygen. There is also the component of when you're doing the exercises, you also put pressure on the blood vessels. So as the muscles contract, they actually compress the blood vessels. When they compress the blood vessel, it means you are cutting off oxygen glucose supply to the cells, and therefore they have to utilize what is already in the cells. Muscles usually store glycogen, so now glycogen becomes the immediate source because the glucose is not coming in, the oxygen is not readily coming in, so now we have to utilize the store that is already inside the cell. So in summary, just like I had before, with the anaerobic pathway, you have the glucose, remember, coming from the glycogen, because at this point, usually you are compressing and cutting the blood supply to the muscles, and therefore we really have to rely on what is inside the cell. So what is inside the cell is the glycogen stores, which have to be broken down to glucose. From glucose, they have to go through glycolytic pathway to release the 2 ATPs. But because you don't have oxygen, you'll end up having pyruvic acid and then lactic acid. This will be released to the heart, kidney, and liver. Now, how long does this one last? I say that this one can go up to 1 minute, slightly less than 1 minute, 30 to 40 seconds. Again, depending on your training. It can go longer for those people who are trained to survive this longer. I think I've already done that. When peak activity, you don't need the aerobic cycle. We've already discussed again about the aerobic cycle. This produces about the 95% of ATP within the cells. Why? Because it is taking the 2 pyruvic acids, taking them through the Krebs cycle and generating all these subsequent amounts of ATP. It utilizes the glucose coming from the glycogen. So at the end of the day, you are depleting the glycogen stores. The problem is once you deplete these glycogen stores, where are you going to get the source of energy? This is when now you start utilizing the fatty acids. And the fatty acids plus, what are they called? The proteins. Before you go to proteins, you have to exhaust the free fatty acids that are available. And that's why you find individuals always have to have ATP. And this ATP, whether they are undergoing through starvation or not, remember that when the person is going through starvation, they do not really have a lot of blood glucose available. The muscles do not have a lot of blood glucose that is being supplied. So they have utilized all their glycogen stores. And now from there, they start utilizing the free fatty acids. And that's why you are told the more exercises you do, the less of the glycogen you have, you'll end up having to utilize your free fatty acids. And the more you utilize the free fatty acids, the more you're likely to lose the body fat. Because you find that whatever you have in store is being used to generate ATP. Now, if you burn up all the fats and you don't even have the glycogen stores, you find that your body still requires ATP. And that's why now you move from glucose as the source, you move from fatty acids as the source, and you go to the proteins. Once you start utilizing amino acids for breakdown, it means that now your body starts to emaciate. So by then, you start even losing body muscle. So you find that although they are available using the aerobic pathway, these can actually provide the energy. But these are used as a last resort after you have utilized all the others. Now, for endurance exercises, when you're using this aerobic pathway, it looks like you have a limitless source of substrate. So for that reason, it can even last longer than what we had initially. So if you are continuously supplying your muscles with oxygen, you keep supplying your muscles with oxygen, then what you'll end up doing is you're continuously producing carbon dioxide, and you can produce ATP continuously. For that reason, you can last like Kipchoge Keino. He was who's? The Kipchoge guy who ran the other day. You can run for 2 hours. I think for us, we can run for more than 2 hours continuously. This is because we are using oxygen as a source of energy. That brings us to now what I was talking about when you have those endurance exercises. When you are utilizing the slow fibers. Slow fibers are using aerobic pathways. And for that reason, they can allow us to continue doing those activities for a prolonged period of time. On the other hand, when you utilize the glycolytic pathway, then this needs a burst of energy supply for a short period of time, which will allow you to provide a large amount of ATP, but only using the glycolytic pathway. So what does that mean? You will first utilize all these pathways. You remember when you were using either the oxygen coming in, burning up the glycogen, oxygen coming in, burning up some fatty acids, but it will reach a point whereby you don't have oxygen coming in and you have to switch to anaerobic respiration. At the point at which you have to switch from aerobic to the anaerobic respiration, you call that point the anaerobic threshold. Okay? So anaerobic threshold is the point at which the muscles have to convert from utilizing aerobic pathway to utilizing the anaerobic pathway. So this diagram, if you have forgotten all the notes that we have discussed today, this diagram captures all. You have to do either you have the short exercises, just go to the gym for a short period of time, or you have a sprint for a given period of time, but this does not last long. You exhaust very easily. As opposed to the endurance exercises where you can run marathon for hours. So if we are to look at these periods of time, when you have that dash, the sprint, within the first seconds, the first 6 seconds to start off the sprint, you are utilizing the ATP that is already in the myofibrils. However, by the time you are doing your 100-meter dash, then you are taking your next 10 seconds to utilize the stored ATP in the form of phosphocreatinine, which is converted to ATP. Now, by the time you are finishing your 100-meter dash, you find that now you are utilizing the glycogen that is already stored in the cells. And this glycogen is being broken down to glucose, and it is the one that is providing for the anaerobic pathway so that you can generate that massive amount of ATP. However, when you go for longer distances, then you find that even your breathing is less labored as compared to when somebody is dashing. When you're dashing, you can even hold your breath so that by the time you finish, you are not using so much energy for breathing purposes, but it will only last a few seconds until you reach the end, and then you have to start breathing. I'm not saying that you always hold your breath, but I'm saying that most of the time you can even last because your focus is in finishing the task within a very short period of time. You can hold your breath and finish the task. And this is very prominent in case of bodybuilders, those who lift big loads, or they are competing for lifting weights. You find that for them to generate that massive contraction, at times they won't even breathe. They hold their breath so that they can focus all their energy to the muscles. The problem with that is that they'll end up having a buildup of lactic acid, and then they have to have an after exercise event whereby they have to get rid of the lactic acid. This is as opposed to somebody who's been running for 2 hours. They find that even their breathing is different. It is regular. It is less labored. It is evenly spaced. so that it continues supplying oxygen continuously to the muscles. For that reason, you find that even when this person finishes the race, their recovery process is not as intense as this other person. Why? Because they were already having oxygen. So probably they will have, because you already had a quickened heart rate, so by the time you're finishing, probably because of that fast respiratory cycle, you also have to slowly go back to your normal breathing and cardiac cycles, as opposed to this one. So we'll come to what happens after the exercise. So before we go to that, let's look at muscle fatigue. What leads to this muscle fatigue? First, the definition will be, when you have muscle fatigue is when you have the inability to contract those muscles despite the continuous stimulation. A very easy way to remember this is when I tell you to stretch out your hand, and when you stretch out your hand, I put a weight, a small weight that you are able to hold. 2 kgs, 1 kg, it doesn't matter. So these are your biceps here. So what you'll do is remember, if you do not sustain this contraction, my 2 kgs will drop. So I have to have sustained contraction. I have to have sustained stimulation to this hand. Keep stimulating, so that you have this isometric contraction whereby the length of the muscle does not change. And if it doesn't change, it holds my weight. I will keep doing this until a point whereby I will feel my hand cannot stay contracted. And what will happen is immediately your hand or your muscles will relax and the hand will drop. At that point whereby, despite you are trying to sustain the stimulation, the muscle just relaxes on its own. That is the point of fatigue, whereby despite continuous stimulation, the muscle just relaxes. What causes the muscle to reach that point? So currently, they say it is because of ionic imbalances. Initially, it was thought that it was just because of the lactic acid buildup, but they realized through experimentation that at times you may even have very high quantities of lactic acid, but you are able to tolerate it, and the fatigue doesn't come in. Although you can say that it will still set in faster than person who has no accumulation. For that reason, we can say the lactic acid contributes, but it is not the main or the only reason. So there are other reasons that can lead to muscle fatigue, and these are the ionic imbalances, whereby mostly you look at calcium concentration. Number 2, we also look at potassium concentration. Then we have the inorganic phosphates. Now you find that all these, if you don't have inorganic phosphate, you cannot have ATP generation. If you don't have ATP generation, you can't have all the functions of ATP, from the sodium potassium pump, from the calcium pump, from the formation of the cross bridges. You cannot have ATP for generating the glycogen stores. You cannot have ATP for phosphocreatinine formation. So if you don't have inorganic phosphate, then you don't have a source of ATP formation. Calcium, if you deplete the calcium, again, you do not have enough for sustaining the contraction. And number 3, you need potassium. This will become important during the action potential. So if you deplete your potassium, again, you have an issue. Usually, our sodiums are very high. Now, the problem is, if you prolong exercises for a very long period of time, remember that the SR is always having those calcium channels, and you need to stimulate the calcium channels to open so that you can have calcium flooding the cytoplasm. Now, problem is, at times if you keep sustained or prolonged stimulation, you're going to interfere with the regulation of the proteins involved. Remember, they are proteins. You can downregulate or you can upregulate them depending on what you are doing. So if you start doing, you are going to interfere with calcium regulation and release. And that will also can lead to fatigue. Now, the problem is, at times, because of this interference, you may also have excessive calcium released into the cytosol. So by the time you are trying to return the calcium back to the cytosol, you may still have a little of this calcium flooding the cytosol. Now, at the beginning of the lecture, I don't know who asked, could this something related to muscle cramps? Now, those muscle cramps are those continued muscular contraction without your stimulation. So you don't tell the muscle to contract, and all of a sudden you see your bulk of muscle starting to contract continuously, and it is usually very painful. Now, that has also been attributed to excessive amounts of calcium, and this could also be attributed to muscle fatigue. So if you have been continuously fatiguing your muscle and you're not allowing it to regulate the calcium stores the way they should, you find that after some time you end up developing these muscle cramps. So there is somebody who was asking about ATP, the relationship of ATP, and in this case I think you are asking, when you have tetanus, continuous contraction, I think, no, then it doesn't fall. This one is talking of lack of ATP. So according to this, you can never have lack of ATP in the muscles unless the person is dead. Then when they die, or even like in case of, like they're talking of severely stressed muscles. So before we go to this extent, you have exhausted all these other sources. But even then, you still find that the cells have some ATP. When you completely lack ATP, is now the situation where we go to perigamotis. So what happens after the exercise? So I told you when I gave you the diagram with the two ladies who are rushing, one was continuously breathing, and you find that at the end of the exercise, because they had a continuous source of oxygen, they are not as exhausted as somebody who did a 100-meter dash. For that reason, you find that their recovery is much faster. They don't need as much oxygen to go and replace what they had utilized when they had no oxygen. On the other hand, for somebody who did a 100-meter dash, they had a buildup of pyruvic acid, a buildup of lactic acid. This lactic acid goes to several tissues. Now it has to be reconverted to pyruvic acid, and when it is reconverted, the pyruvic acid has to go through the Krebs cycle and finish the process that had been initiated. Krebs cycle will require a lot of oxygen. For that reason, after somebody does that dash, they have to do a lot of panting so that they can take in the extra oxygen, which will replace what they had utilized without oxygen. So this is what has always been referred to as the oxygen debt. So when you are running, you are generating ATP and you're not using oxygen. You are creating an oxygen debt. You are building up your debt for oxygen so that immediately you finish the exercise, you have to inhale, take in more oxygen than you normally breathe in so that it can go and create more or burn up the pyruvic acid, creating the ATP. And what will that ATP be used for? It will replenish the stores. You remember you depleted all your stores. So now that ATP will be used to replenish it. It will replenish the phosphocreatinine because you had burnt it all. You find that because of that, you have that excessive intake of oxygen at the end of the exercise period. Questions so far? So I think this will be the last slide that I had on sources of energy. So I don't think I'm going to go back to skeletal muscle because we've utilized all the way up to which types of muscles do we have, what energy do they have, what sorts of energy are they utilizing, how do they initiate contraction, how many types of contractions do we have. So I think with that, the next two topics I'm going to focus on are cardiac muscle and smooth muscle. I'm still inviting questions. Before I take attendance. Questions before I take attendance. Yes, Samuel.
My question is related to tetanus disease.
Yes.
When we were studying the receptor pathways, I think there is somewhere I've seen that bacteria which causes tetanus, uses I think it's G protein receptor pathways. So I wanted to understand maybe how is the connection with that receptor pathways and the calcium ions, which causes the contraction of muscles, the sustained contraction of muscles, which is tetanus. Yeah, like that.
So what it will do is that it will cause continuous stimulation. You remember when we talked about ionotropic versus metabotropic pathways or stimulation? Okay.
Yes.
So with the metabotropic, it utilizes the it utilizes the G-coupled receptor pathway. So if you bring in a toxin that is going to interfere with that pathway, it means when you stimulate and you're supposed to initiate the termination of the stimulation, but the termination does not occur. So what happens? You continuously stimulate the cell. And when you continuously stimulate the cell, what happens to the calciums? You release them from the stores because it is stimulate the neuron stimulate the motor end plate, stimulate the sarcolemma, release calcium. So now, if you have a continuous release of calcium, then your muscle stays continuously contracted. You see the connection now?
Yes.
Okay. So patience.
Yeah, I'm wondering, like, I thought, okay, I've not got the clarification about the muscle fatigue, but again, I got a question, because I thought muscle fatigue was caused by lack of energy or ATP, but you said it's those calcium and iron imbalances and some and all those other factors. And I'm like, sometimes when you are exercising, you get tired like in 5 minutes, and then after like you persist and keep on pushing, you almost forget is are the ions, are those ions now have then reoccurred in that person exercising, or it's psychological? Like in short, can the ions I don't know how to phrase this question. Can they, like in the middle of your exercise, after you are tired and then you feel like you're no longer tired, is that the ions are now being imported into your muscles, or it's just because you have forgotten about it?
Okay, so you're able to see my slides?
Yes.
There are two slides I didn't show you. The general definition of fatigue, the decreased capacity to work and to reduce efficiency of performance. So like you're describing, there is that tiredness. Yeah, you are doing work and you get tired, and it could be psychological. Just like you have defined, it could be due to muscle activity. Now, muscle activity, we talked about the ATP, but generally, we said this is the last thing that happens. Before you go to ATP depletion, you'll have a buildup of so many things. There is also a fatigue that can occur at the synapse. Remember that the synapse, we have the release of acetylcholine. Acetylcholine is normally stored in form of vesicles. So if you continuously stimulate a neuron, it will continuously lead to release of these vesicles, and you don't have enough time to build up more acetylcholine. So when you deplete your source of acetylcholine, even that muscle can undergo fatigue. So general fatigue can be due to all these types or all these causes. Now, the one that I'm talking about, the muscle fatigue, is the one that is due to the muscle itself can no longer stay contracted, and the reason being because of what you have. You have like the lactic acid, you have buildup of electrolytes, you have all these the different electrolytes that are present there. So as you say, when it comes to the issue of fatigue, it is not a really straightforward A or B or C. It's a combination of factors. At times you wake up in the morning, you have not done any exercises, you haven't done anything at all, but you feel so tired. So why are you feeling tired? Now that is where you bring in another component. Could it be emotional? Could it be even like when you don't have enough sleep? You wake up in the morning and you feel fatigued. So like I say, it is not a one dress fits all. It's a combination of many things. Okay? So does that answer you, Patience?
Yeah.
Okay. So I wanted us to quickly go through what is remaining Let me see. Because I always refer back to my, what are they called? To my course outline. So generally, if you look at what we are covering on muscle physiology, we've looked at the machinery of how contraction takes place, neuromuscular. Are you able to see my screen?
Yes.
Okay. So we've covered structure and function of all those contractile proteins, neuromuscular junction. We've done reflexes. What are the definition, the different types of reflexes, their importance. We've done isotonic, isometric contraction. When it comes to contraction, remember when we are talking about the motor units and the speeds and all that, I think there is the one part that I have not really touched on about the curves, but for power output, I think we have touched on that. So now what is remaining is we need to look at smooth muscle and cardiac muscle. So far we've done the structure. So even my quiz should include this. We have looked at contractile activity in skeletal muscle. Even my quiz too should include that. However, I have not covered cardiac and smooth muscle contractile activity. So I will not include this in my quiz too, but it will definitely be in the exam. Now, because of that, this contractile activity of smooth muscle will also be captured here, and it will be talking about the large state. However, I hope you're noting or you're recording this. When we are comparing smooth muscle, cardiac muscle, skeletal muscle, we talked about their action potentials, their activities. So I will include that. How can you compare an action potential of a contractile cell of a cardiac muscle as compared to the action potential in a pacemaker cell? How does that compare to a pacemaker cell of smooth muscles? And how do those action potential compare to the neuron? So that one should be included. If you have an issue with it, talk to me before quiz 2, because I will include that. I remember I did it as a pre-lab session, so it is not in this course outline, but it is part of the pre-lab because we needed to understand those the electrophysiology of these tissues so that you can understand what was happening in the lab. So I'm going to quiz you on that and everything else that is week 8 to week up to today. Let's say up to today, because I'm not going to talk about contractile activity of smooth and cardiac muscle. So next week, hopefully, I'll finish on that, and that will be week 11. So that week 12, I'll finish off with what is the large stage. How do you couple excitation, contraction in the smooth muscles? And after that, if you need me to answer any question, I can help you out before week 13. So do you have any question? Yes, better.
Okay, so during when we're talking about the sympathetics, you say that the digestive system is inhibited.
Yes.
But then when you are working out, like they usually say you should have a heavy meal like 2 hours before and not eat a heavy meal, let's say 30 minutes close to a workout. So if in case you eat usually during, let's say 30 minutes, a heavy meal, you
Yeah.
get almost a sense of nausea when you're in the gym. But then it's almost like you don't get that during those fight or flight responses, even though the digestive system is inhibited. Why is that?
You get nausea, that's what you're saying when you do the exercise.
Like when you do the exercise, let's say maybe 30 minutes, you get a heavy meal. And then
Yeah.
after 30 minutes, when you do the exercise, it's almost like you feel a sense of nausea and uncomfortable anyway.
Now, you'll find that I think the next topic, the next course we are supposed to be looking at is respiratory and digestive physiology. And during the digestive physiology, you're going to see that food lasts in several parts of the digestive system at a given period of time. So in your mouth, it can last for so long as you voluntarily keep it in the mouth. But once it goes to the esophagus, it takes only about 10 seconds before it hits the stomach. And now in the stomach, it can last for maybe a period of up to 3 hours. So imagine if you have just fed and then you start doing exercises after half an hour. So where is your food located?
Yeah,
In the
it's in the,
stomach.
yeah.
So you start feeling like you want to throw out because you are squeezing on the stomach and you're not allowing. Remember, when you're doing those exercises, you've inhibited the digestive system. So you've inhibited to allow the food to go into the intestines. You've prevented it from going. It is stuck in the stomach, and yet you're squeezing on the stomach. So the food will only look for its only way out, which is through the esophagus. And that's why you
Okay.
start feeling nausea. Now,
Okay.
you are told to do that after about 3 hours. The food will have left the stomach. It will have gone into the intestines. Now, you find that between the intestines and the stomach, there is a sphincter that holds the food in the intestines and it doesn't go back. If the food is already within the intestines, then you inhibit the intestines. What will happen is they will be contained there. They cannot move out, and therefore you cannot feel nausea and you cannot throw up. But at the same time, they are not being digested. So they're just keeping in wait until you finish. And then when you finish, they continue with their process. Does that answer your question?
Okay. Yeah.
All right. Any other question? So what I will do, yes, I do have, I will take attendance. Give me 1 minute.